In this video, we're going to continue talking about the transfer of proteins from one place in the cell to another. So in the previous video, we talked about proteins that are synthesized in the ER. So after synthesis in the ER, any transmembrane, secretory, or lysosomal proteins will go to the Golgi apparatus. The Golgi has 3 to 6 functional membrane sacs called cisternae. So we have the cis, the medial, and the trans. And proteins will enter in the cis, get modified, and exit out of the trans. And there are two different methods for transportation. If you're transported through the Golgi in membrane vesicles that butt off of one compartment to the next, as we can see here, we call that anterograde transportation. While if you're a passenger in Golgi compartments as they become different components, going from cis to trans, we call that cisternal progression or maturation. Or you can actually go backwards, and we call this retrograde transport. So here we can see some cisternal progression, and you could go the opposite way, as kind of being shown here. This is retrograde transport. Now, the things that are transporting our proteins are called vesicles, and they are coated with proteins, so coated vesicles, not coasted. And on the outer faces of these vesicles, we have specific proteins that make our vesicles. And we'll talk about those specific proteins in a minute here. And these proteins are flexible scaffolds that will promote the formation of our vesicles. And the vesicle will butt off whatever protein it came from and fuse to its target membrane. And so this will actually preserve the orientation of our transmembrane protein. So the way to look at this graph is that the vesicle is heading towards the cytosol, and these purple are your membrane proteins. So you can see that we have some carbohydrates in the lumen. Well, what happens is that the vesicle hits the cytosol version of your membrane and it will start to fuse. And these proteins, since they're in the lumen, they will actually be facing the outside. And eventually, the whole vesicle fuses and becomes part of the membrane. So now your transmembrane protein, the part that was in the cytoplasm, stays in the cytoplasm or the cytosol. Now we know of 3 different types of coat vesicles, so 3 types of proteins. You have your Clathrin, which form a polyhedral framework around the vesicles. These go from Golgi to plasma membranes. COPI, which is fuzzy coating for both anterograde and retrograde transport. And any ER resident proteins that escape the ER, COPI vesicles transport these proteins from the Golgi and back to the ER. This protein is also known as coatomer. And COPI transports proteins from the ER to the Golgi. Again, COPI2, so COPI2 will then return any COPI coated vesicles back to the Golgi. And COPI2 is made out of 2 conserved protein hetreodimers. Hetero means different, dimer means 2. Now we're going to look at Clathrin to understand our vesicles because they are the best studied type of vesicle protein. And Clathrins are made from proteins known as Triskelions. You have 3 heavy chains, which are the blue here, and 3 and bound to 2 light chains. So heavy chains and our light chains. And these will form cages. So here the red is one triskelion, yellow and green are others. And the light chain actually inhibits your heavy chains coming together like this. So they form a regulatory rule, so you're not forming vesicles without needing to carry something. And these triskelions are flexible, and that allows for different sizes and shapes of vesicles to be formed. Triskelions and Clathrins will also participate in endocytosis. which was engulfing a specific protein from extracellular medium to interior. So they help with that process as well. Now, thinking back to ER resident proteins, most proteins that belong in the ER will have the same 4 amino acids at the C terminus. This is K, D, E, and L. Yeasts have H, D, E, and L, but most proteins will have KK and something else, or 3 Ks and then 3 something else's. So proteins that reside in the ER, they can accidentally leave the ER through a vesicle. So here we have our rough ER, and one of our proteins, whoops, got in the COP2 protein that was going to the Golgi. Now, once in the Golgi, we have coatomer, which is COP1 again. This will bind to the lysine in our K-del sequence, and so it will bind any ER resident proteins in the COP1 vesicles, which are going back to the ER. The Golgi also has these K-del receptors. So these K-del receptors bind the K-del sequence, binds the proteins that belong in the ER. These K-del receptors go into COP1 and back to the ER you go. So anything that escapes and is not meant to escape will be returned to the rough ER because of the 4 amino acids at the terminus. And that's it for this video. I will see you in the next one.